confocal microscope, or a laser scanning microscope. This uses a laser beam to scan objects. It assembles the image bit by bit, almost pixel by pixel, onto a computer. It can produce high resolution, high contrast images, so more than we can get with a light microscope. It allows us to see structures at different depths of a specimen. So you know when you kind of scroll through the focus of a light microscope, you can maybe see something that is 3D and you can see different bits of it, and some bits will be in focus and some of it will be out of focus as you go through it. This actually allows us to see all of those parts of the 3D kind of structure using this laser. And so we can see whole living organisms and cells without it being kind of all blurry, bits of it being all blurry and out of focus. So here's a picture that's been taken with the laser scanning microscope. So you can see we've obviously stained different parts, different colours, so we can use the computer to colour the image. So the cytoskeleton is red and the nucleus is blue here. So we've obviously done that using the computer. But you can see the level of detail. You can see the detail in the cell membrane around the edge. You can see the cytoskeleton, which you wouldn't have been able to see with a light microscope. And these are really helpful for use for things like medical diagnostics. So being able to really quickly see a sample under a microscope and be able to kind of look at it in sort of 3 dimensions, see all this detail and be able to decide what it is. Also for biological research. So to be able to look at live cells in enough detail where we can see this level of detail, but they can be living and moving around and carrying out processes is really, really useful.